So good evening, everyone. My name is Seemanti Aditya. I am the Ph.D. PMRF scholar in the Department of Biotechnology at IIT Madras. And so I am a teaching assistant for this course. And today we'll be talking about the week 11 content of this course. We are almost nearing the end of the NPTEL course. And so today we'll be talking about autoimmunity and graft rejection and self-tolerance, and how is all these autoimmune diseases, why are these autoimmune diseases so common nowadays, and a lot of people are getting affected by rheumatoid arthritis and this diabetes, type 1 diabetes mellitus. So all these diseases, these are belonging to autoimmune diseases. So what is autoimmunity? So basically autoimmunity is like an inappropriate response of the immune system against self proteins. So any kind of your self antigens or your proteins which is expressed on your cells, if that proteins are recognized by the immune system as a foreign particle or a foreign pathogen, then the immune system is going to elicit a response against that self antigen, and that is going to harm your own body or harm your own organs and cells, and that is known as autoimmunity. So it can involve the activity of both the humoral and the cell-mediated components. So humoral means the antibody component, and the cell-mediated means the T cell component or the adaptive immune responses. So both the antibodies and the T cells are associated with autoimmune responses. And there are many examples of autoimmune diseases which you can like you might have heard about. For example, the rheumatoid arthritis, Graves disease, and type 1 diabetes. These are like really common autoimmune disorders nowadays. And so autoimmune responses, why do the autoimmune responses take place? So basically there is still a doubt, like why like irrespective of, there are so many selection when your B cells and T cells are maturing, or when your B cells and T cells are getting developed in your the lymphoid organs, and when they are getting matured in your thymus or bone marrow. So instead of, like, there are so many pathways where the self antigen, those cells which are interacting with your self antigen, those cells are eliminated or deleted, and they are led to the apoptotic pathway. So in spite of that, there are cells which are going to interact with your self antigen, and they are going to cause these autoimmune disorders. So this is basically not a very common practice. It does not happen in all individuals, but some individuals, they have a greater tendency to have a higher immune response, and therefore autoimmune responses are very prominent in those individuals. So some terms associated with autoimmunity or autoimmune responses are self-tolerance. So what do you mean by self-tolerance? Basically, all the mechanisms that can avoid the improper activity of immune system against the self components are termed as tolerance or self-tolerance. So mechanisms like, deleting those antigens which are, deleting those cells which are interacting with your self antigens, or inhibiting immune responses against your self proteins. So those kind of mechanisms or processes which are happening in your body in order to avoid this autoimmune responses, so those come under the term as self-tolerance or tolerance. So basically it can include elimination of immune cells that show self-reactivity, and also active inhibition of immune responses against self proteins. So we have studied that during maturation, that we need some kind of cytokines, we need some kind of chemokines which are also suppressing the immune system, which is responsible for inhibitory effects, which are responsible for maintaining the balance between an overwhelming response of the immune system and and moderate response of the immune system. So whenever those kinds of processes are getting affected, whenever those inhibitory cytokines are not produced in a good amount, whenever your cells are being somehow escaped from the negative selection and positive selection during the maturation of B cells and T cells. So those kind of times it leads to autoimmune disorder, and it leads to the escape from self-tolerance. So your cells no longer can recognize your own like it can no longer differentiate between self and non-self proteins or cells, and therefore it leads to autoimmune disorders. Like that. So yeah, so there are certain ways where this tolerance is mediated. So there is basically two points, or uh, two two regions in your maturation of B cell and T cell pathway, where tolerance is exhibited by your the cells which are going to get matured. So one is known as the central tolerance, and the second is known as the peripheral tolerance. So by the name you can figure out that where this tolerance is basically exhibited. So central tolerance takes place when your B cells as well as T cells, when they are just getting developed from the lymphoid precursor or the hematopoietic stem cells in the primary organs, say the bone marrow and the thymus. Bone marrow and thymus for T cells. So your lymphoid precursor comes from hematopoietic stem cells. So hematopoietic stem cells, from there your lymphoid precursor cells develop, and then from your lymphoid precursor cells, different types of your lymphocytes will develop, both B lymphocytes and T lymphocytes. So here what happens, your both B lymphocytes and T lymphocytes are exposed to the medullary cells, B cells or T cells, which have on their surface self antigen. So there are self antigens which are expressed on the surface of your cells present in your bone marrow and thymus. So these are the cells present in bone marrow and thymus. So these cells basically express on their surface self-antigen. So these self-antigens are, exposed to all of your maturing B cells and T cells. So these are your maturing B cells and T cells in your in your bone marrow or thymus. And so these cells are, recognizing all these self-antigens which are already present in the bone marrow or thymus. So there are 10 cells which are developing, and each of the cells have different kind of receptors. And so this kind of, so some receptors will recognize the self antigen, some receptors will not recognize the self antigen. So there will be three categories of your cells in the central tolerance, where one set of clone will not recognize any of the self antigen. So those clones will mature. So maturation of clones which are not specific for self antigens are present in the primary lymphoid organ. So this set of clones mature which do not recognize any of the self antigen. Second set of clones which are very specific for the self antigens, they will be deleted. So they will undergo apoptosis. So this set of cells, these are recognizing self antigen, and they are having very high affinity to the self antigen. So those set of cells will die by apoptosis. And the third set is, there are clones which are which have kind of some kind of affinity with the self antigen, but they can suppress the self antigen recognition. So these are the regulatory cells, or Treg cells. So Treg cells are cells which have moderate affinity to your self antigen. So moderate affinity to self antigen means that if given a chance, they might also in the future might interact with your self antigen and cause autoimmune disorder. So because of that, these T regulatory cells, they upregulate certain kind of genes which help in converting such clones which have this moderate affinity to self antigen into T regulatory cells, so that these T regulatory cells will like in the future they do not cause any trouble. So moderate affinity to self antigen clones, they will help, they will have the ability to suppress that self antigen recognition in the future due to the upregulation of certain genes. We will look into how T regulatory cells do that, and how what genes are upregulated in T regulatory cell, which will inhibit the self antigen recognition. So this is the first step, where it is where in the bone marrow or thymus, your cells are basically selected or selected for not recognizing the self antigen. So here, after crossing this barrier, when the cells are reaching your secondary lymphoid organs, say spleen and your lymph nodes, where they are exposed to the pathogens from the external surface, which is not self antigen. So here these are mature lymphocytes, and these lymphocytes will have access to bind to foreign antigen, and this is known as peripheral tolerance. So this takes place in secondary lymphoid organs. Okay, secondary lymphoid organs. The secondary lymphoid organs include spleen and lymph nodes. So here what happens, that those cells which escapes this kind of central tolerance, and where one of the clones, say, they do not get recognized, like they are recognizing the self antigen but they are still maturing into the mature cells. So those lymphocytes, when they are coming in contact with foreign antigen, so this foreign antigen, they will be recognizing foreign antigen as well, because sometimes what happens is the foreign antigen has epitopes which are very similar to your self antigen, and therefore those cells will act as an effector cell against those foreign antigen. And the set of cells which are binding to self antigen and which had escaped this kind of self central tolerance, those cells will again undergo another type of tolerance mechanism, where if it is binding to self antigen which is present in your secondary lymphoid organ, say in your spleen and lymph nodes, so cells of your spleen and cells of your lymph nodes will also have certain kinds of antigen, self antigen or self proteins. So these cells, if they bind to those self antigen or proteins, they will either die by apoptosis or they will show energy. So what is energy? Energy means unresponsiveness to the antigen, or your foreign pathogen. So this is not going to react, this is not going to mediate any kind of immune response. So either it will undergo apoptosis or it will become unresponsive. So this is known as energy, the term is known as energy, or it will turn into a regulatory T cell. So T regulatory cells, if it is binding to a self antigen. So this is known as peripheral tolerance, where deletion, energy, or regulation of lymphocytes that can recognize self-antigen in the peripheral tissues. So there are two sets of tolerance. One is central tolerance and one is peripheral tolerance. So here what happens, again, just the B cells and T cells during their maturation, either in the bone marrow or thymus or in the secondary lymphoid organs, they are selected for the cells which do not interact with self-antigens. Only those cells will mature into effector T cells or effector B cells, which will mediate your adaptive immune responses. So regulatory T cells which inhibit self-reactive cells, what happens is, so there is this thymocyte. Okay, this is growing inside your bone marrow. This is growing inside your, say, thymus. So after getting matured from the bone marrow, it is moving towards the thymus. And so in both bone marrow and thymus, what happens? There will be three sets of clones. So here it is mentioned 3 sets of clones. So each set is exhibiting different properties. So this thymocyte, one set has low affinity for self-antigen. So this clone will basically mature into effector T cell. So this is an effector T cell. The other two sets, what happens is one is having a high affinity to self-antigen. So this set will die. So this will die by apoptosis. And the one which is having intermediate or moderate affinity to the self-antigen, this clone will upregulate the expression of another gene which is known as FOXP3. So this is a transcription factor, upregulation of a transcription factor. So the cells, the T cells clone, which are recognizing self-antigen with moderate affinity, those clones will show upregulation of FOXP3 transcription factor. So upregulation of FOXP3 transcription factor will lead to the conversion of this type of clones into another set of cells, which is known as the regulatory T cells. These are the regulatory T cells. These cells can then act on the normal cells which have been matured into the effector T cell. So this regulatory T cell can suppress the reaction to self antigen. So these cells also produces certain cytokines, certain interleukins, and etc. So these cells will have the property which will further inhibit itself. It will inhibit the T regulatory cell to interact with self antigen. Also, it will suppress the activity of the normal T cell to interact with self antigen as well by producing certain cytokines. So T regulatory cells has a very important function in suppressing the reaction to self antigen, both for itself as well as for the cells that did not initially show any kind of self antigen interaction. So what would lead to this T cell showing some kind of self antigen interaction? So there might be certain mutations in the T cell receptors or certain mutation in the B cell receptors that could lead to this particular cells which initially did not interact with self antigen to show self antigen interaction. So that time those cells will might mediate the autoimmune responses. So that time T regulatory cells will play a very important role to suppress those immune responses or autoimmune responses. So T regulatory cells helps in inhibiting these autoimmune responses. So how is the autoimmune responses mediated? How the autoimmune responses by the antigen presenting cells? So what are antigen presenting cells again? Antigen presenting cells can be like dendritic cells or the B cells or the macrophages. So all these cells, they can also mediate some kind of autoimmune responses, because autoimmune responses are mediated both by the adaptive as well as the innate immunity branch of your immune system. So autoimmune responses are mediated both by the innate immune responses as well as the adaptive branch. So in the innate branch, what happens is that there might be certain cells like dendritic cells or the macrophages and B cells again, they are part of both the adaptive and innate. So dendritic cells, they have on their surface MHC class 2. So all the antigen presenting cells have on their surface an MHC class 2, and they are presenting all this processed antigen to the T regulatory cells. T regulatory cell, basically this is a subset of T cells only. So all the T cells recognize antigen which is processed and presented by MHC class 2. So similarly, T regulatory cell will also recognize antigens which is processed and presented by MHC class 2. So apart from inhibiting certain cytokine production, so it is recognizing the antigens. This might be self antigens. So this self antigen which is processed and presented by MHC class 2 to your T regulatory cells, there is another kind of receptor interaction, co-receptor interaction, that is CTLA4 from the T regulatory cell, which interacts with CD80 or 86 from the dendritic cell. So this is B7, and so CTLA4 B7 interaction basically. So this interaction, so this entire interaction, which is mediated by both the MHC class 2 presenting the antigen to your T cell receptor. This is the T cell receptor, and the CTLA4 CD80-86 interaction. So this interaction basically increases the production of IDO. This is an enzyme which is helping the this is immunodeoxy 2,3-deoxygenase, and it helps in the helps in the conversion of tryptophan to quinine. And quinine is a immunosuppressant. So this acts as an immunosuppressant. So because of the production of quinine, due to the interaction between this T regulatory cell CTLA4 and CD80-86, there will be increase in the production of IDO enzyme, and this IDO enzyme will help in the production of quinine, and this quinine will act as an immunosuppressant. And because of the higher amount of this enzyme, this molecule, there will be inhibition or less production of other kind of cytokines like IL-6, TNF alpha. So TNF alpha, IL-6, these are also this also mediates inflammation and inflammatory responses. So due to the inhibition of the production of TNF alpha and IL-6, it will lessen the effect of the self-inflammatory reactions or hypersensitivity reactions. Also, it will inhibit the autoimmune responses from the antigen presenting cells. So inhibition of all these cytokines, inhibition of the production of inhibition of the cytokines and the production of all the immunosuppressive agent is responsible for the inhibition, responsible for providing protection against autoimmune responses by the T regulatory cells. So T regulatory cells plays an important role for mediating the suppressing the autoimmune responses by both the innate part of your immune system as well as the adaptive part. So there are in spite of all of this presence of T regulatory cells and central tolerance, peripheral tolerance, and all of those, there are still certain diseases which are taking place in your taking place regularly, and we can hear a lot of cases. So there are certain diseases, examples are given over here, and what are the antigens which are responsible for all of these diseases. So this is a table which shows some of the autoimmune diseases in the humans, and you can see that there is Graves' disease, where the thyroid stimulating hormone receptor is affected, and it is due to the autoantibody against the TSH receptor. Also, there is type 1 diabetes mellitus, which is affecting the pancreatic beta cells. So type 1 diabetes mellitus is a very common type of disease which is increasing progressively among the population nowadays, and there is a lot of individuals who are affected by type 1 diabetes mellitus, and it is affecting the beta cells, where the insulin secretion gets affected, and your body has higher amount of glucose, so there is hyperglycemia, and so it is like it kills the body slowly, because it also has certain side effects on kidney and functioning of the kidneys. So type 1 diabetes mellitus primarily is due to the antibodies against the beta cells, pancreatic beta cells. Also certain T cells which will affect this, which will show some inflammatory reactions, as well as the mediate cytotoxic properties to the beta cells and kills those beta cells. So there are numerous such cases where the autoimmune responses take place. For example, the rheumatoid arthritis, where the connective tissue is affected of the antigen. Connective tissue in the sense your blood is affected. There is like, this is systemic autoimmune disease. Okay, so there are two types of autoimmune disorders. For example, the organ specific autoimmune diseases, which is affecting certain organs or the cells of particular organs, and there is systemic autoimmune diseases, which is affecting the blood or your entire body. So entire body, and this is organ specific, and this one is organ specific. So in systemic autoimmune diseases, the antigen or the autoantibody against your self antigen is present in your blood, and it will affect it will affect the joints everywhere in your body, because blood can reach your joints from any part of your body. So that is the systemic and organ specific autoimmune diseases. You can go through this, and what are the different kinds of effect, like what are the different effector mechanisms, and what are the self antigen and target gene. Could be you can just go through all of this just maybe once, and you can once just take your like eyes through this, and you can understand that what kind of effect this kind of diseases can cause. So apart from that, there are certain causes for autoimmune disorders. So there could be like infection, like viral, bacterial, fungal, where different kinds of the antigenic epitopes, which are similar to the viral pathogen or bacterial pathogen or fungal pathogen, is like whenever there is an infection, say viral, bacterial or fungal, so because one antigen can have several epitopes. So say this is an antigen, and this is having epitope 1, and this is having epitope 2, also epitope 4, 3. So there are several epitopes. Now, during antigen processing and antigen presentation, some epitopes could be similar to your self antigen. Say epitope 2 is having similarity with your self antigen. So this will result in antibodies which will target this particular antigen. So initially, say during your viral, bacterial or fungal infection, the antibodies are generated against this epitope, which is actually viral, which is actually bacterial or fungal. But also due to like similar kind of epitopes which is present in your self proteins or self antigen, your antibodies which were generated initially for this particular antibodies which are generated for this particular antigen will interact with self antigen as well. So because of that, your autoimmune responses, and so one of the causes can be infection. Second cause is toxins. So smoking or dyes, iodine, etc. This can lead to a greater inflammatory. There could be an inflammatory reaction, and this inflammatory reaction might trigger certain mutations or certain like certain kinds of, like if there is smoke, if you are smoking, and then there could be certain particles which are like present in your blood, and against that inflammatory reaction is taking place. And so during that inflammatory reaction also, antibodies might be produced against those particles which are which comes in your blood due to all of this like exposure to toxins. So that time also your inflammatory reactions as well as the autoimmune responses can take place. Also there is stress that could be psychological, oxidative or metabolic, or ultraviolet light and endoplasmic ER stress that we talk about. So all of these reactions, all of this stress also is responsible for developing this inflammatory and autoimmune responses. Also food habits are also one of the major reasons for the autoimmune diseases. If you are having a lot of junk food or a lot of food which is basically increasing the reactive oxygen species or ROS levels in your blood, so this is also is very very deleterious and harmful for your body, and that will generate the responses, immune responses against this particular species, and that is going to be harmful in the long run. So all of these reasons can be the causes for autoimmune diseases. Apart from that, there are also certain genetic causes that is always there. So genetic causes are also associated with autoimmune diseases. So certain kind of genes of the cytokine receptors, or the certain kind of chemokine receptors, all of those genes, if it is mutated or if it is affected, if it is overexpressed or if it is downregulated, those could also be certain like causes for the autoimmune diseases. So that we do not know yet that what could be the reason for so many autoimmune disorders coming up, but still there are researchers who are working for this, so that we can understand it better and get a treatment which could provide protection against these diseases. It's like no such like treatment is known for properly cure this disease. So it is like we need to study more about these diseases. So one of the examples for autoimmune disorder is myasthenia gravis, and myasthenia gravis is because of the acetylcholine receptors which are being attacked. So what are acetylcholine receptors? So acetylcholine receptors are present in your nerve endings, and it helps in transmitting the signal from your transmitting the nerve impulses, and helps in muscle contraction or activation. So what happens is that there are antibodies which are generated against these particular receptors. So initially, if there is no infection, if there is no autoimmune responses, your the nerve endings will have acetylcholine. Acetylcholine is a neurotransmitter. So these acetylcholine molecules, so these are acetylcholine molecules, this is a neurotransmitter and it transmits signals. So acetylcholine will be binding to the acetylcholine receptors, and then it will mediate the signals, and it will signal the muscle for contraction, and so muscle will be activated. If the acetylcholine receptors are already bound with certain antibodies, say acetylcholine antibodies and autoantibodies against acetylcholine receptors, then all of these receptors will not be available to bind to the acetylcholine. Although the signal is coming from the nerve endings and the acetylcholine is released, still the acetylcholine will not be able to bind to the acetylcholine receptor, and muscle activation will be inhibited. So that is one of the reason why in myasthenia gravis people will undergo paralysis, or people will not be able to show any kind of movement, because the acetylcholine receptors are destroyed due to the binding of autoantibody against acetylcholine receptors. So myasthenia gravis, binding of autoantibodies to the acetylcholine receptor will block the normal binding of acetylcholine and subsequent muscle activation. In addition to that, the anti-acetylcholine autoantibody will activate the complement pathway. So complement is another pathway for the innate immune responses, and this complement is also associated with a greater inflammatory responses. So these complement proteins are also binding, and it is causing deleterious effects in the pathogenic in the pathogens, where it creates pores, and then it kills the pathogen. But here what happens is it activates the complement pathway, which is going to mediate a greater inflammatory response, as a result of which the muscle end plate. So greater inflammatory response means a lot of reactive oxygen species is generated, and a lot of cells are coming into like DCs are coming, uh, macrophages are coming to your site of infection. And so here the muscle cells, basically they are targeting the muscle cells, and the muscle cells get damaged. So as a result of which the muscle cell will undergo injury, and muscle cell will ultimately die. So muscle cells injury is taking place, and it dies. So as the disease progresses, basically the acetylcholine receptors also declines, as well as the muscle cell gets injured, and the person will undergo paralysis. So myasthenia gravis is one kind of autoimmune disorder. uh, so apart from that, so this is how different cells, like basically the T cells, transfers allograft rejection. So there are so many types of graft. So we can know. So there is autograft, there is allograft, and there is xenograft. So Autograft means when certain tissue is taken from one part of your body, tissue taken from one part of your body, and transplanted to another part. Allograft means tissue which is taken from a different individual, tissue taken from different individual and transplanted to another individual, and transplanted to another individual. And there is xenograft. So xenograft means like it is the basically tissue is taken from different species and transplanted to a different species. Also, there is isograft. So isograft means this is 1, 2, and 3, and 4. So isograft means when the tissue is taken from identical twins, or identical twins basically. So genetically similar kind of organism. So that is identical twins only. So here there was an experiment which was conducted to see that how T cells are mediating this allograft rejection. So what happens is there is a mouse, and this mouse is they had transplanted a skin graft from another mouse. This is known as strain A mouse. And so this mice, after 14 days, it shows first set rejection. So your immune system takes certain time to recognize the foreign transplant or the foreign graft and shows rejection. So first set rejection is shown after 14 days. So here the tissues undergo necrosis. So after a certain time, when the same mouse is given a second skin graft from again the same individual, the second set of rejection takes place in a very smaller amount of time, say 6 days. So initially it took 14 days to reject this tissue. In the second graft or the second skin graft, it took more or less time. It took just 6 days to show the rejection. So same experiment was done here. What they did is the mouse strain which had already the strain A graft, strain B mouse which had a strain A graft, they took the splenic T cells from this particular individual. These T cells were injected in the mice which already had the graft from strain A. So this mice showed second set rejection. So instead of instead of just directly transplanting the skin graft for the second time, if they are transplanting the splenic T cells for the second time, it is showing the similar kind of reaction, just like the one where two skin grafts are transplanted in the same mouse. So this shows that the T cells are basically mediating the allograft rejection or the graft rejection, because when you are taking the T cells and injecting it, it is also showing a second set rejection, and it is rejecting this particular first skin graft that was present. So this is similar to, like, this is the experimental demonstration which shows that T cells are actually transferring the allograft rejection. Also, certain autoantibodies, if are produced against this strain A graft, those antibodies which are those antibodies also, if you take and inject in strain B mouse, that mouse will also show some kind of rejection. So both the antibodies as well as the T cells are responsible for allograft rejection, but in this particular experimental demonstration, they show that T cells are, transferring the allograft rejection. So there are certain effector mechanisms which are, associated with allograft rejection. So, for example, the antigen-presenting cell, which is, which is interacting with your graft. So this antigen-presenting cell is interacting with your so this is interacting with your graft, and it is processing all those proteins present on the surface of graft, and this antigen is processed and presented by MHC class 1 to the TH cell, and then TH cell will mediate the responses through the different cytokines which are produced, and these cytokines will again, lead to the production of different subsets of T cells, the T helper cells, the cytotoxic T cell. Also, it will help in the differentiation and maturation of B cells, and also the T DTH cells. So this will help in this particular set of cells, which will again go and interact with the antigens present on the surface of your graft, and this will generate a very wider immune or autoimmune responses, wider autoimmune responses. So this is an exaggerated immune response. So from just a single APC that recognizes an foreign graft or foreign protein from the graft and presents it to the T helper cell, and this T helper cell will help in mediating this cytotoxicity as well as the MHC expression. There will be this MHC expression which is recognized by the T helper cells. Also, the macrophages will be activated, which will produce lytic enzymes and kill these cells in the graft. Also, it will phagocytose the cells in the graft. It will cause membrane damage. It will cause lysis of the cells. It will cause antibody dependent cell cytotoxicity. So all of this is mediated by the antigen presenting cell, which is actually presenting the processed antigen to the T helper cell, and then finally helping in the maturation and growth of all other cells that are interacting with the graft and showing allograft rejection. So this is also one type of responses against this is a response against a foreign graft that had been planted in the individual's body. Also, the antibodies that are associated with the graft, those antibodies are also responsible for the rejection of any kind of graft. So not only just the T helper cells, so both the T helper, this is the adaptive part of the immune system, so T helper cells, so adaptive part of the immune system, this is known as this is also the adaptive part, but the antibodies part, this is the cell mediated part. And this is the humoral immune response. So the graft which has been transplanted from the donor, so this graft already has certain antibodies. So these are pre-existing host antibodies which are present in the graft, and these antibodies will bind to the antigens which are present in the capillaries. So this is the capillaries which are present in the host's body, and these antibodies will then activate the complement pathways, because these antibodies which are present in the host in the donor's graft will have certain kind of similar responses, because this is not a specific kind of response, because complement proteins, they recognize the FC part of the antibody, FC region of the antibody. So this is not any kind of specific immune response. So this antibodies is going to activate the complement pathway by non-specific activation, and this complement pathway will then produce all this. Complement proteins, they will generally get activated by by dissociation into two components, say A part and the B part, and this will then result in the greater immune response by attracting neutrophils that will release lytic enzymes, as well as the inflammatory responses by secreting lot of cytokines and lot of chemokines, etc. So these antibodies will activate the complement proteins. Complement proteins will then, show a higher immune response by attracting the neutrophils as well as secreting cytokines. And these neutrophilic lytic enzymes will destroy the endothelial cells or the platelets which are adhered to the injured tissue, causing vascular blockage. So basically, all this is present. This is inside the kidney graft itself, where this complement protein is attracting all these neutrophils. These neutrophils are, killing the cells, and they're destroying all the cells present inside the kidney graft. Because neutrophils as well, they are not very specific. These are the innate part of the immune system, and so these are killing all these kidney cells, and these kidney cells are then getting all these capillaries. The capillaries get blocked by all the cells that are killed inside these capillaries, and as a result of which there is a blockage again, and the kidney graft will not work normally. So the rejection of kidney graft, or the rejection of any kind of graft for that matter, is caused by both the humoral response, the cell-mediated response, as well as the innate immune responses, where the antibody is also very important for recognizing the foreign antigen, as well as the innate immune system also plays an important role in creating this huge inflammatory response, and huge mediates huge killing of this foreign cells, by not foreign cells, the graft cells, by the non-specific mediated immune responses. So the graft rejection is again both mediated by humoral as well as innate, as well as the cell mediated part. So that is all about what I had to say. So now we'll just go through the questions. So you might answer them. I'll just... Okay, so a lot of people, not so actually what happens is that the answer is not A. So it's mentioned that after exiting the thymus, mature T cells are subjected to the secondary selection, where majority of the self-reactive T cells are deleted or lost their reactivity. So secondary selection is not the central energy. Central energy takes place when there is primary selection, when there is the cells are not mature T cells, they are immature T cells, and they are present in the bone marrow. They are not, they are not mature enough to be going to the secondary lymphoid organ. So when they are exiting the thymus, they are basically going to the secondary lymphoid organ, and secondary lymphoid organ will not mediate central energy. So it's B. Yeah, so the answer is peripheral energy. Okay. Then which of this is not an autoimmune disease? So this is a easy question. Right. So most of everybody has given the correct answer. The answer is AIDS. So AIDS is not an autoimmune disease. It's a viral disease, retroviral disease, basically. And it's caused by the human immunodeficiency virus. Okay, SFJ, does energy and tolerance is the same? No. So tolerance means your self antigens, like your the cells which are interacting with your self antigen, they are basically deleted. They are not in the circulation, or they might be suppressed. They might be turned into a T regulatory cell. That is known as tolerance. But energy means that even if it is reacting, even if it is recognizing certain self antigen, it is unresponsive, so nothing will happen to it. Neither it will be dying, neither it will be turning into a T regulatory cell. So it is basically a dormant cell. So that is known as energy, where it is not responding, it's not responding to any kind of signals or any kind of immune response. It's not going to elicit any kind of immune response, or it is not going to do anything. But tolerance, it is going to either the cells which is interacting with the self antigen, it will die, or it will be converted into a self regulatory T cells. So so there the energy and tolerance is different. Okay, SFJ. So this question, immunologically privileged sites. So what do you mean by immunologically privileged sites? Uh, so basically, immunologically privileged sites means where the immune cells are not very active, and even if there is certain pathogen, it could be neglected by the immune cells because they do not have access to those pathogen. So most of the cases in brain, eye, and testis, they are immunologically privileged because they are not very accessible to the immune cells. They are not very accessible to the immune cells. So any kind of infection which might have occurred over here, it's not going to be cured by immune cells. So the correct answer is liver. You have given the correct answer. Yeah. So what is this? This is basically, it should be a gap over here. Hemolytic anemia. What's hemolytic anemia? It's very like the term is also very self-explanatory. Like it is should be associated with RBCs only, right? Because anemia is associated with RBC, sickle cell anemia, or any kind like hemolytic anemia. So this is another kind of autoimmune disease which is associated with self-destruction of RBCs for reasons unknown. Still, people do not know why RBCs are being destroyed. So it might be due to an infection, might be due to certain drugs which are affecting the proteins present on the surface of RBC, and immune cells are attacking the RBCs. So whatever is the reason, it is still not known, but it leads to the destruction of RBCs, and RBCs are reduced to a very less number in the body, and as a result of which the oxygen transmission is affected, and the affected individuals are they turn sick, and they do not have enough oxygen reaching the tissue. So the correct answer is the destruction of RBCs, or the option A. And the last, okay. Does the autoantibodies only secreted by escaped cells from selection process? Autoantibodies, yeah, actually, all during the selection process, all the cells which are, even interacting with the self antigen, they will be released due to certain mistake of the immune system, and those cells will only produce the antibodies against the self antigen. So yeah, so those escaped cells are basically producing autoantibodies, correct? SFJ. Yes, Rajkumar, you have a question?
Ma'am, good evening, ma'am.
Good evening.
Ma'am, 10th assignment one question doubt, ma'am. Eighth question explain, ma'am.
Eighth question, 1 second. Okay, so the question is, a number of cytokine receptors signal via the JAK-STAT pathway. Which among the following statements is incorrect? Okay, so JAK kinase gets activated on dimerization of cytokine receptors. JAK kinase phosphorylates receptor sites and creates docking sites for STAT molecules. STAT, upon binding to the receptor, gets autophosphorylated. Okay, STAT dimerizes and translocates to the nucleus where the gene expression takes place. Okay, so JAK kinase gets activated on the dimerization of cytokine receptors. It is correct. JAK kinase phosphorylates the receptor sites and creates docking sites for STAT molecules. That is also correct. STAT, upon binding to the receptor, gets autophosphorylated. So STAT does not get autophosphorylated. JAK is a kinase, right? So okay, let me go back to the slide of last class slide. So week 10. Second, I'll share the screen. Did I not? There is a JAK slide. Okay. So basically, you can see that in this JAK-STAT pathway, what happens is there is this cytokine receptors. So the question is like, what is correct and what is incorrect? So the option number C is incorrect, where it is written that STAT autophosphorylates itself. Instead, what happens is there are cytokine receptors, alpha subunit and beta subunit, where the alpha subunit is recognizing the ligand and the beta. So alpha subunit and beta subunit, when it comes together, it gets dimerized. And so dimerization will activate the JAK pathway and JAK-STAT pathway. So JAK kinase is activated, and JAK kinase, it's a kinase, so it will phosphorylate itself, all the tyrosine residue present in the JAK. So this blue colored region, it is phosphorylating itself, and this is JAK is autophosphorylating itself. So JAK kinase gets phosphorylated, and here it will help the STAT molecule to come and join this region of your receptor, and JAK will then help in phosphorylating the STAT. So tyrosine phosphorylation of STAT rather JAK kinase is phosphorylating STAT. So the option number C is mentioning that STAT is autophosphorylating. So that is incorrect. Basically, JAK is helping in phosphorylating STAT. So that is why the option C is incorrect. Understood?
Okay, ma'am. Thank you, ma'am. Ma'am, answer C option right, ma'am.
What?
Eighth question.
Huh?
Answer is C. C or A,
Yes.
ma'am?
C, C. Answer is C.
Okay, ma'am. Stat upon binding receptor autophosphorylation.
Yeah. Yes.
Okay, thank you, ma'am.
Autophosphorylation. Correct. Yes, yes. Okay. So that's all. Any more questions? Okay. So if not, we'll meet again in the next class in Friday. I hope that will be the, I think that will be the last class. Okay. So thank you all. I hope you are learning and you have learned quite much about immune system and immunology. We'll meet again in the next class. So thank you. Have a nice weekend.